Hello, I'm Heng Leung, a surgeon scientist.
And I'm Jackie Stockley, a postdoctoral researcher.
We are based at the Beatson Institute for Cancer Research and the University of Glasgow. We want to introduce you to our recent publications in the BJU International. In this report, our early investigations based on a single human prostate cancer cell line suggests that autophagy may have a pro-survival effect as an adaptive response to treatment. We propose that future preclinical and clinical investigations in this area are warranted.
Despite PSA testing, many patients still present with advanced prostate cancer. For these patients with incurable disease, androgen-ablation and chemotherapy are commonly used. Our initial data looking at androgen-ablation with or without docetaxel chemotherapy revealed an important interaction between the two therapies. We confirmed that docetaxel is highly toxic to all of the prostate cancer cell lines tested, but interestingly, the LNCaP cell line, which had been chronically depleted of androgens, the LNCaP AI cells, and then acutely exposed to docetaxel treatment, was substantially more resistant to docetaxel when compared to the control parental cell line. Importantly, however, when androgen is reintroduced to the LNCaP AI cells, which are adapted to growth in androgen-depleted environments, we observed that docetaxel-mediated cell death was significantly enhanced. We know that the androgen receptor is specifically involved in this sensitization, as this observation was abolished by the anti-androgen bicalutamide.
Autophagy can be considered as a homeostatic process for incorporating cellular components into large vesicular structures called autophagosomes, which will then merge with lysosomes to degrade their contents, typically generating products for energy or recycled as building blocks. Autophagy has therefore been suggested from previous studies to play a potential role in cell survival and may also be assisting resistance to chemotherapy treatment. Our recent research has shown that activation of androgen receptor can in fact delay the induction of autophagy, despite serum starvation of prostate cancer cells. Now, this finding led us to ask the question whether the converse may be true, that is, whether the suppression of androgen receptor is itself a sufficient stimulus to induce autophagy. In addition, given our data on the combined androgen ablation and chemotherapy treatments we heard just now, we wanted to test the significance of autophagy following both androgen ablation alone and combined with chemotherapy.
The status of autophagy was studied in the LNCaP cells, and as expected, when the LNCaP cells were exposed to the anti-androgen bicalutamide or Casodex treatment, both expression of the androgen receptor itself and the PSA levels were significantly reduced. The presence of autophagy was demonstrated by the expression of the microtubule-associated protein LC3 in a punctate pattern and elevated levels of its lipidated form detectable by Western blot analysis. Using a pharmacological means to inhibit autophagy by 3-methyladenine 3-MA, cell death was significantly enhanced when autophagy was suppressed upon androgen ablation with or without chemotherapy treatment. To our knowledge, our data reveal for the first time that inhibition of the androgen receptor with bicalutamide actually induces autophagy as a pro-survival process.
So in summary, based on our early data reported in this manuscript, we would suggest to the reader that future in vitro and in vivo studies are warranted to test the therapeutic importance of manipulating autophagy. Thank you for your interest in our paper.